So just going to quickly read out a little bit about Andrea's talk and a little bit about Andrea as well. Yeah, if you like, you can do that. Andrea's talk tonight is entitled Shorebirds and Working Estuaries, Evidence-Based Identification of Critical Habitats, Food Webs and Management Pressures. So Andrea's talk is about the Hunter and Port Stephens estuaries supporting internationally important populations of migratory and resident shorebirds. And they also underpin major industrial, port and community activities. So Andrea will present a summary of a 4-year research program conducted by her team at the University of Newcastle's Centre for Conservation Science, which is focused on shorebird habitat use across 3 working estuaries. So the results reveal hotspots of shorebird usage within the estuary and visitation of key roosting sites by foxes, while also revealing differences in the diet and site reliance across species, highlighting that some species are more dependent on specific food pathways than others. So Andrea, trainer in marine biology, specialising in animal behaviour, animal physiology, neuroscience and ecology at the Universities of Lausanne, Geneva, I probably said that wrong, did I? That's good. And Zurich, in Switzerland. She completed her PhD at Macquarie University, followed by a Swiss National Postdoctoral Fellowship at McGill University in Canada. So she joined the University of Newcastle in 2005 as an Australian Research Council postdoctoral fellow and earned a lectureship in the School of Psychology in 2009. She is currently an associate professor in the School of Science, where she is the senior leader of the Centre for Conservation Science. Andrea is also a HBOC member and a passionate advocate for local shorebirds, and in her spare time is also helping organise the Australasian Shorebird Conference, which is to be held here in Newcastle in September this year. We should all go along to that. Okay. So thank you, Andrea.
I have to also say that I have had the great pleasure of being Louise Williams' PhD advisor. There you go. So thank you everyone, thank you very much for coming. This is actually a talk that I gave for about 20 government partners last week, but a shortened down version because Mick said I had to go really fast. So that says I would also like to acknowledge the traditional owners of the land where we work, in particular the Warramai people and the Awabakal people. They are the traditional owners of the land where we work and we live and we play. And I deeply respect, and my team deeply respects their connection to the land, and we pay our respects to their elders past and present. I would also like to thank the amazing students who work on these projects with me, with my advice. They challenge me every day, they make me learn every day, they make me a better person every day, and it's really such a pleasure and honour to work with these young people. They do all the hard work. Here it is. I don't talk about everyone's work today, but these are the main people, and we've got a few additional names down there. I'd also like to thank the amazing team at the University of Newcastle. So this work is very, very interdisciplinary. We're asking lots of different questions way outside my core expertise, and it wouldn't be possible without the academics who are in what we have now, the Centre for Conservation Science. And if any of you are interested, I do have a little booklet here about the centre at the University of Newcastle, and they bring such skills, it wouldn't be possible without them. I also want to, of course, thank the Hunter Bird Observer Club, because the work would also not be possible without this amazing community partnership that we have. They bring enormous ornithological ground-level expertise to the project and really teach me new things every day from their advice and from their wisdom. And of course they bring A-class banding and allow us to catch and tag birds. So I'm very pleased that we're able to produce, and we all agree we need to disseminate this work, and we have this sort of co-authored publications that are coming out of this, and it's lovely to do this work with them. And I also want to thank Glenn McFadden, who's here in the room, because I have to say that some of the work we do is extremely difficult, and I will come back to that in a minute. And if it wasn't for Glenn, who's sitting in the back of the room and runs a kayaking tour group, the small company that takes trips into Fullerton Cove, we would not be able to access Fullerton Cove. So the work would not be possible, I'm talking about today, would not be possible without Glenn. So this is, thank you Glenn, this is an opportunity for
I'm going to talk about shorebirds.
Look, the place I usually start is to make you aware that Australia is an incredibly coastal country, the most coastal dwelling country of one of the most coastal dwelling countries in the world. In 2019, 87% of Australia's population lived within 50 kilometres of the coast. It's phenomenal. We used to be all concentrated in cities, but now we're spreading across the coast since COVID-19. There's been a tendency to leave the big urban centres and to spread. And of course, we share this coastline with all these amazing species of coastal shorebirds. There are residents, there are migrants who do these enormous interhemispheric journeys twice a year. I'm sure you're all very familiar with them. What do they have in common? They are generally reliant upon intertidal habitat, and many of them are in very strong decline, which I'm sure you're all aware of. And we all agree, mostly, that habitat protection is absolutely key to the long-term survival of shorebirds. Now, protecting habitat is quite complicated with shorebirds. There's many species, but shorebirds are highly dependent upon networks of sites. Sites, locations that contain both roosting sites and foraging sites. The roosting sites have to be above the tide, so it's subtidal habitat, and the foraging habitat is usually intertidal habitat. So at high tide, they rest and roost in these high tide, above the high tide, and then low tide, they disperse across the landscape and they go to forage in these habitats that become available when the tide goes out. And then they shuttle between these locations. So they shuttle between roost sites and intertidal sites every 6 hours. And they do this day and night. Right? These sites, they depend upon these sites, this network, right? You wipe out one of these sites, a roost site without a nearby intertidal flat to feed is useless. Vice versa, an intertidal flat where you can't make them somewhere rest when the water comes in is useless. The other aspect is these sites have to be relatively close to one another so the birds can fulfill their daily and seasonal energetic needs. So they can't be kilometres and kilometres apart. I will make this point again at one point in my talk. The other part that is less thought about in this network of sites is that there could be habitat that's contributing to the quality of the network. We don't necessarily see birds, but is necessarily important to the integrity of the network. And that could be something that, for example, is a habitat that feeds the invertebrates that live in the intertidal mud. Okay? And I'll come back to this point. And it's well recognised now, it's well accepted now in sort of environmental protection law that to protect shorebirds we have to protect the network. The problem with this is that the network precisely makes it difficult to protect because a proponent can develop one of these sites and say, look, it doesn't matter, there's another roost site over there. The birds can just move, right? That's not a great disturbance. The problem with that is that you have to do good surveys. So you have to do good surveys at the site, and if you don't do the sites at the right time of day or you don't do them in the right season, you might find there are no birds and think, oh, it's not an important site. So if this network of sites can kind of be eroded by the gradual degradation of site by site, always thinking, well, there's another one over there that the birds can just move to. So we have to understand how the birds use this network, cross-tidal and circadian cycles, so day and night, to really make sure that we can protect the whole thing, and that when a proponent proposes to do something like dredge a port, for example, and that might impact one of these sites, we can think about really how we compromise the integrity of the network. But each site can be whittled away, one decision at a time, and so your whole network can collapse. That's the problem. So I'm going to present 3 pieces of work today, and I'll call them 3 studies for the sake of it. But the first one is about trying to understand chasing abundance among species across sites within the Hunter Estuary and across years. The second piece of work is trying to understand site use within the Hunter Estuary across tidal and day and night cycles. And the third piece of work is asking about this question about whether habitats outside this network of sites where we see the birds is actually important, even though we might not see so many birds using it. So this is the Hunter Estuary. I'm sure you're very, I don't need to explain the Hunter Estuary. It's wonderful. It's one of the most important shorebird. It's a very important shorebird site, both nationally, but also, the most important in New South Wales. Many thousands of birds use it every year. It's heavily managed for shorebirds. So there's micro-clearing going on, there's tidal flow management, and there's been restoration of wetlands with shorebirds in mind, trying to create habitat for them. And these all require ongoing management. And Hunter Bird Observer Club has done an absolutely tremendous job at doing high tide roost counts for over 25 years, which is just extraordinary. So we have an amazing amount of work of data that has been collated at the estuary level mostly. And this is work from Alan and Anne in recent papers looking at how these counts have changed across time by species, and you can see that depressingly a lot of these lines are going down. Now, there are nevertheless some gaps, and the gaps are in the spatial and temporal coverage of the survey work. So, these are high tide daytime counts. So this network that I showed you, they're using this network all day, all night, and they're using it to forage and to roost. With this data, we know where the birds are at high tide, and we know where they are during the day, because that's where we can find them. But what about the rest of the network? How are they using it? Now, this is not a criticism, right? This is not saying, oh, there's a gap here. It's actually really hard to do this work, right? You have to go, the birds disperse at foraging times, at low tide, they disperse, you have to go and find them. So literally you're scouring the landscape, looking for where the birds are, right? Now try that on a mud flat, right? Mud flats are really hard to access at low tide, and without Glenn here in the room, we wouldn't be able to do what I think is actually quite dangerous work, right? Going into a mud flat that is soft, where you sink up to about here in the mud, and you try to get yourself out of it again when the tide comes back. And now, that's difficult enough, but try doing that at night, right? Even high tide roosts are difficult at night, leave alone foraging sites. So this is really hard work, right? So it's not surprising that it presents challenges for conservation, but we have to still try and do it. So the first piece of work I'm going to talk about is work by Mattia Taylor in her thesis. We decided that we were going to take a look at these high tide counts that have been done over this long period of time in the Hunter, and instead of collating the data at the whole estuary level, we were going to look at the different sites where the birds are counted at high tide during the day. And really the areas that concentrate on the count sites, we really just concentrate maybe on Kooragang Island, Ash Island, Stockton Sandspit, Tomago and Hexham. Those are kind of the main areas that we look at. I'm not going to tell you about methods, okay? I'm just going to put some graphs up and show you. I have to trust me, happy to take questions about methods. You do a lot of model fitting, a lot of stats, you get the statistician to help you, so you make sure you're doing this properly. So you get the experts in the room, but wherever you look, whatever site you look at, whichever species you look at, things are going down, right? Why is it interesting to look at how, some people might think, well, what's the point of looking at it? Because what can you tell about the reasons why the birds are declining or they're changing in a particular site? It might be the birds are changing up in the LAC, right? That's why they're decreasing. They're just fewer are coming to the estuary, and so of course they're decreasing here. So what's the point of looking at site level data? Well, one of the interesting things is that, true, a species can be declining because there are fewer individuals coming to the site because some problem is happening up in the LAC. But within the estuary, what you could find is that in some places a species is going down, consistent with the global trend, but in other places in the estuary it's going up. Now that can tell you something about, well, what's happening at those two sites. Clearly one is becoming preferred and the other one less preferred. So differences in these patterns across sites can tell you something about what's happening. Similarly, a species can be declining, but if it's declining really strongly at one site and much less strongly at another, again, it tells you, well, there's something about this site where the birds are continuing to go, right, despite the overall number decreasing. So relative differences can tell you something. So back to these figures, most of them are going down. However, interestingly, I can tell you that moss lapwings are increasing in Hexham Swamp, which is one of the restored sites, which is interesting. And the red-legged avocet are increasing at area E, which is also an area where the tidal flow has been restored. So that's interesting, despite the fact that things are decreasing elsewhere. I mean, these are species that are increasing in the estuary, so this is fairly consistent. What I'm showing you now, and I've highlighted in red this box, is one interesting, very interesting pattern emerged from this analysis that I was actually not expecting, I have to say. Look at these figures in the middle. So we've got Tomigo, we've got Hexham. So these are these restored areas where the tidal gates have been opened, water has been let back in, and look at these curves. They're all curved. They go up and they go back down. We could not fit lines to these things. We had to fit them as curved, right? So the species come in, and then they go across. They go again across time. So you probably can't see at the back there, but we've got about 20 years on the x-axis. So across a 20-year period, we're seeing these curved lines. So what's happening? The birds are, well, one good thing, the birds are clearly coming, right? When you let the tide back in, you change the system from a freshwater to a saltwater system, the birds notice this, which I think is a great thing. It tells us when we do something, birds find it. How on earth they find it, I don't know, but they find it. They're saying, this is great, here we go. Then the next question is, why did they change? Why did they go again? Why did they stay? That is entirely consistent, actually, with ecological, lots of ecological work that says that what happens is the system will transition through to a new state, and in this transition phase you can get lots of species coming because there's lots of things changing, and then it will restabilize and you will lose species again. So I think what it raises is the question of what is Tomigo and Hexham now like? How can we change it such that these species stay, this remains a good foraging ground or used site for them to use rather than these decreases? So it raises lots of questions. And then we have some interesting things. Golden plover are also increasing in Phoenix Flats, so these are the curved relationships. And two interesting things are happening over at Fullerton Cove Beach. Also an area there on the east of the Fullerton Cove, on the east area. This is an area that's been cleared of mangroves. So it was recently set back up a little bit like Cody Point. And you can see really interesting, again, the birds are responding, right? They were on the decrease, but now marsh sandpiper and common greenshack are increasingly using this site. So this is really important to say, wow, we really need to keep removing the mangroves on this. This is becoming an increasingly important site, despite the fact that common greenshank are decreasing at area E. So clearly area E, tidal flow has changed something, they're not so keen anymore, but hey, this area over in Fullerton Beach, that is now becoming good for them. So we can go and ask questions. What's great about Fullerton Cove? What's the difference with area E? Why don't they like it over there? Why does this species not like it there, right? So I don't have any answers, but lots of questions. The second piece of work is moving in to try and understand a little bit more about where the birds are going at low tide, right? This challenging phase of activity to understand. So this is work that's in Louise Williams' PhD. Louise went out and did some surveys across both the Hunter Estuary and also this time Port Stephens Estuary. I put the habitat, they're interesting estuaries to compare. This one is salt marsh mangroves in the Hunter Estuary, I'm sure you're aware. Up in the Port Stephens, it's a much more marine, steep-edged estuary. There's not much intertidal zone because the sides go down quickly, right? So you can see a very thin edge here, but much more marine. It's got some seagrass over there. Hunter doesn't have seagrass, right? So you have this sort of riverine influence over on the west, and then a more marine influence on the east, which is interesting to compare. And when I started this work, nobody knew where the birds forage. We had some inkling in the Hunter because it's kind of known, because it's been studied for a long time, so there was some inkling. But over here in Port Stephens, I had no idea. I did a massive piece of work for the Nature Conservancy where I basically I did 8 really, really big full-on surveys in the estuary where I drove with a boat around the estuary 8 times looking for birds. Funded by the Nature Conservancy, thank you. And I saw that Pindimer was really important. There's some islands that are really important down here, but these are the 3 sites that I proposed to Louise to go and look at. Pindima, Swan Bay, and Soldiers Point. So, these figures show you the maximum count. On the x-axis, we have all the different sites, and in black are the hunter sites, and then in grey, I'm going to show you the Port Stephen sites in a minute. And Louise divided the birds up into migratory, resident, coastal obligate, and generalist. So, migratory and coastal obligates you find in Bartel Godwit, Forest and Curlew. Your generalist you find more your golden plover, and then you've got your resident species like the red-necked arrowsbill and pipesbill. Now, really interesting, where do you find the birds at low tide? She went to have a look at low tide, okay? Where do you find them? Well, this is where they are. You find them in Fullerton Cove, Curringane Dyke Ponds, and Stockton Sands Bed. This is where you find them. They're not over here in these other locations at low tide, okay? Pretty consistent pattern, even amongst the residents, Fullerton Cove, and also Stockton Sands Bed to some extent. They're there at low tide, okay? So lots of sites, but restricted use at low tide. So this is just to say you don't find them over here at low tide. So look, we've got a great network, but at low tide you find them over at these two sites, not the rest. And this is just some more detailed data with Eastern Forest and Curlew, Bartel and Godwit and Mars Lapwing, and you can very clearly see once you zoom in on these boxes that this is where you're finding the birds, the maximum counts at low tide, with the Mars and Lapwing showing a lot more generalised use across the sites. What about Swan Bay? Well, Swan Bay, 3 sites surveyed. Where do you find the birds at low tide? You find them where the bars are, which is basically Swan Bay and Pindima. Okay, so birds, Soldiers Point, not so much so, perhaps somewhat in the, actually even in the residents, but perhaps in the coastal obligates a little bit more. Sorry, where am I here? Stockton Sands Bed. Oh, I put this bar is in the wrong place. Yeah, here. So coastal obligates, generalists, basically Swan Bay and Pindima are the 2 most used sites at low tide. Now, what you can ask is the birds are there at low tide, well, what are they actually doing at low tide, right? We assume they're foraging. Well, it turns out, so this is the percentage of birds that are foraging. So you're watching, say, 100 birds and 20 of them are foraging, or you're watching 100 birds and 99 of them are foraging. Okay, so the proportion of birds that are foraging. In the Hunter Estuary, where are the birds foraging? Well, you have to have bars that are really high up on these figures. Generalist, coastal obligates, and migratory species. They're only foraging in Fullerton Cove and Curringane Dyke Ponds. They might be at Stockton Sands Bed at low tide, but they're not foraging there. Okay, so now you have an even more restricted set of sites that's being used to forage for these birds. The residents are more distributed in their foraging, activity. Okay, so this really suggests that these 2 sites are very, very important for foraging. And in Port Stephens, here bars are higher for both Swan Bay and Pindima, so the birds are not only there at low tide, they're also foraging there at low tide. Okay, so you can see now, perhaps slightly less with the generalists and residents, but both sites are used for foraging. Where you find them is where they're eating in Port Stephens. Now, we did a very quick kind of genetic analysis of the sediment in these locations, so Dyke Ponds, Fullerton Cove and Stockton Sands Bed. We ran an essay over this for invertebrates, and these are really just showing you what's called an alpha diversity measure, so it's essentially a measure of the diversity of invertebrate DNA in these samples. Okay? So if it's high, you've got lots of genetic diversity. If it's lower down, you've got less genetic diversity, which just suggests less invertebrate diversity. So fewer species, fewer different species of invertebrate. And you can see here that in those locations where we find the birds foraging, there's greater diversity of genetic diversity of invertebrates in the mud. And then this is just another way of representing this in terms of clustering, how similar the DNA pieces are to one another. And you can see again that Dyke Ponds and Fullerton Cove kind of group together. They're kind of similar in their invertebrate DNA mix, but Stockton Sands Bed is very different. Now these data just suggest that there might be something about the invertebrate communities in these that make the mud more or less suitable for the birds to forage in. That's what it suggests. So again, lots of questions. Maybe it's down to the quality of the mud. So this is the second part of this second piece of work, which I'm going to tell you a little bit about our automated telemetry work. So this is, we've been going for a while. This is now Maxime Marini's PhD. He's focusing on this in his first chapter. So we have set up in the Hunter Estuary an automated telemetry array. What is this? This is a set of stations here you can see in blue. They are fixed radio telemetry stations. So they have an antenna on them. Here you can see this is, you can see at the back there's a tall antenna sitting on a mast, about 9 meters high, the top of it. This is a directional antenna that's, I'll tell you in a minute, it's a directional antenna, really long antenna, and when the bird flies past, we get it, we detect it. So we attach a tag to the birds, very lightweight tag, we glue them on, and we get a few weeks of detections, and when the birds come within these stations, we can tell they're there, because the receiver tells us it detects the tag. So every one of, you can see here, is an omnidirectional antenna, so basically we can tell exactly when the bird is there and when it leaves that site. And then over here we have a directional antenna that's pointing it to the west, and we can tell when birds are flying past it. Okay, it's a little bit different. We're interested in kind of shuttling between what we thought was happening. So the stations are placed strategically. They're not placed haphazardly. Right? They're places where we think would be useful. What is this giving us? It's kind of giving us a map of which species we see where, to start to try and understand the diversity of the species, because they don't all use these sites in the same way. We have a network of sites, but how are different species using all those sites? So we build up kind of a map of who goes where. And so, I got 52 individuals of 8 species. This has taken a long time to accumulate, about 3 years, mostly 3 years of tagging going on. And so we're pretty happy. We've got some nice data coming out of this. This is a really crude measure. It's just the number of individuals detected. Okay, so they have been, while they had a tag on, they've been at least once to one of these stations. So a really crude measure, right? But it already gives you some idea of how many species are going to which of these places. So let's take a look at Curlew Point and Fullerton Entrance. So again, Fullerton Entrance is here, Curlew Point is down there, a well-known high tide roost. We're getting lots of different species using these areas, right? We look at all the different species, they're basically all, at some point, they all come to these 2 sites. Now remember those low tide surveys, right? Those were again, those are the locations that are important. Let's go over to Millen's Pond on Kooragang. Again, quite a lot of diversity, fewer individuals, okay, overall, but pretty good diversity. So lots of species are liking Millen's Pond. If we go to Ramsar Road and Swan Pond, are a bit further over, individuals, but not so much diversity. We've got golden plovers going over there, but not so many of the other species. In Tomago, we've only got 2 species there, bar-tailed godwit and red-necked avocet using Tomago. And windayers that people hadn't surveyed before, that's not usually surveyed, where we thought we might find birds using it. So here on the north side of Kooragang, golden plover, go there, okay? Nobody else so far goes over there, which is kind of a bit weird. It's a nice sight, but let me show you a little bit of what the data look like so you can understand how this works. When these are 2 bar-tailed godwit data, 2 individuals, red and green. They've got a bunch of pulses here because they are at, this is Curlew Point, they are at Curlew Point at a particular time. And we know exactly when the pulse occurred. So what we can do, because we have such temporal accuracy, we can map the tide onto these detections, as you can see, high tide, low tide. And what we can also map is sunrise and sunset, okay? So we can really tell where are they going relative to the tide and relative to day and night. So this bird was at Curlew Point at high tide. And then you can kind of look how this extends across time. You've got days, right? And I've made in grey the nighttime periods and in yellow those daytime periods to help you navigate this figure. But look at what's happening. So these two birds are over here at Curlew Point at high tide during the day. Where are they at the next high tide, which is during the night? They've moved to Tomigo, right? Where are they the next day? The next high tide, they've gone back to Curlew Point. And then the next day, this tide fell off, but the next day this bird's gone back to Tomigo, and then back to Curlew Point, then back to Tomigo, and then back to Curlew Point, right? So they are actually switching from day to night. They're not using the same high tide rates during the day, they won't be counting them, as during the night, okay? And you really see this repeating, so there seems to be something going on. What you can do now with the data is you can take all these pulses and you can calculate a duration presence. You add this, plus that time, plus that time, plus that time, and then you divide by the number of days. And then you can represent that duration by each period. A daytime high tide, a daytime low tide, and the same thing for night. So a nighttime high tide and a nighttime low tide, right? So you get 4 periods, basically. High tide, low tide, sliced by day and night. So you can work out that, and then you can start looking at where are the birds across these 4 periods of time, okay? So daytime high tide, daytime low tide, nocturnal high tide, nocturnal low tide. So the nocturnal ones are in grey, and the daytime ones are in white. So if you navigate this, and then these are all our different species here. And this is the number of hours that we've seen these birds using these sites. So it's incredible data, right? You can see that some of them, Curlew sandpiper, we've got 300 hours. We know where these birds are, right? They're basically on our antennas at some point in time. Let's take a look at the general patterns here. So if you look at golden plover data and Curlew sandpiper, we get good coverage during the day for both species, but not so good during the night, right? Where are they going during the night? We've got less coverage, not so clear, okay? Pied stilt, pretty good coverage both day and night. We know where they are, okay? Forest and curlew, we have very few data. We do actually know where one in particular is. The second one, I haven't quite wrapped my head around the data, but I'll show you the first one. Wilbur, they're not on our stations. Where are they going? They're outside our array, okay? Red-necked avocet and Bartel godwit are interesting. You can see this is daytime high tide, this is nighttime high tide. We have, it looks low, right? But it's still about 50 hours of coverage of these birds. But low tide, not so good, okay? Either during the night or during the day. Now I actually, for both of these species, I'm going to show you the data in a moment, but I'm going to argue that we don't have good coverage of these two species because they're going to Fullerton Cove. And Fullerton Cove is a directional antenna, so they don't sit beside the antenna, they fly past it. So of course our duration is very, very small. Right? So I actually think that we know where these species are at night. I'm going to show you a little bit more data just to give you a little bit more insight here and some take-home messages. Here are the, on the left high tide, on the right low tide, and again, white bars are during the day and dark bars are during the night. So essentially, this is Bartel godwit. Where are they during the night? Well, they're either at Curlew Point, but they're also typically at Tomigo, which is exactly the data I just showed you. So that was a pattern on two Bartel godwit that we got earlier on, that switching between Curlew Point during the day and Tomigo at night. Same thing here, birds, well, we've got more birds now. They are at Curlew Point during the day, and they're leaving at night. At Curlew Point, they're leaving. Where are they going? They're going to Stockton. So these birds use two sites, differentially day and night, and those two sites are Tomigo and Curlew Point. And I think, as I said, I think we would have Fullerton entrance because it's a flyby antenna, we don't get it. Pacific golden parrot, where are they at night? Grey bars, this nice blue put around them to help you navigate. They like Curlew Point as well during the night, like the Bartel godwit. What is the other site they like there? They like Millam's Pond. They come over here at night. They don't seem to want to go to Tomigo. Who knows? They go over to Millam's Pond. So two sites, but not the same ones. Okay? And I also want to show you a few things. Again, Curlew Point is used during the day, for example, at low tide, but they leave There's less usage during the night. Same thing here, more usage during the day, less at night. So where they are during the day does not predict where they're going to go at night. So that's why nocturnal data is so important. Where are red-necked aniset going? Well, they like two sites as well, but not the same. They like Fullerton entrance, so they actually like to sit in front of this antenna, be it at high tide or low tide, particularly to forage apparently. But the other side they like is Tomigo, and they are going there at low tide, possibly to feed. They're going at low tide during the night, and possibly also during the night at high tide, but they're certainly going there during the night. So like the Bartel Godwit, they like Tomigo at night, just like the Bartel Godwit. But their other site during the day is not Curlew, it's not Curlew Point, it's Fullerton entrance instead. Right, so different preference again. And my last one is pied stilt. Where are they? Well, this is going to be simple for you. Pied stilt are easy. They just seem to hang over at Fullerton entrance. Okay? They like it during the day, they like it during the night, they like Fullerton entrance. So we really need to look after that new beach where I showed you the numbers are going up, remember? It's becoming really important site within our network. Now, a couple of slides on this eastern curlew, because it really highlights some aspects about that network. This is one bird we managed. Eastern curlew are terribly difficult to catch. We managed to get a tag on early on. We were so lucky we got a tag on the bird. And these are data we've actually published. I come back to these because I really think it's quite revealing. This is the presence of this bird at Millen's Pond. Okay? Now in red, this is our flyby antenna. So the different colours here are actually two different antennas. You see a brief pulse as the bird flies past that antenna, and over it comes to Millen's Pond in green at the low tide. As the tide goes up, the bird is still there. It's still here, right? And then at the next low tide, it's still at Millen's Pond. So this whole period of low tide, low high tide, and then another low tide, the bird has stayed at Millen's Pond. Where does it go with the next high tide, which is a high tide, a high tide, and you can see here, far greater amplitude, and it's at night, they leave. It leaves, it flies past Fullerton Cove, the Fullerton Beach antenna. So we think it's come back. We didn't have an antenna over there in early days, so I can't tell it has, but I suspect it's shuffled back. What is the take-home message? The take-home message is from this, is that the bird is staying at Millen's Pond despite the low high tide, which means it doesn't shuttle back to Curlew Point at a low high tide, right? Remember I told you, these sites have to be close together. They have to meet their energetic needs. This suggests this bird does not go back to here. We might think, look, it's just over there, it's the crow flies, surely it doesn't take that much energy. The bird doesn't go if it doesn't have to. Clearly, it's a low high tide, so it can still stay there. It's not pushed up into the salt marsh. But at night, when there's a high tide, maybe you end up in the salt marsh, and that's not great for eastern forests and curlew. So off it goes. So this suggests to me that if you can avoid the energetic costs of going back, you do. You don't shuffle with pleasure. The other interesting thing about Motor's data is there's variability in the signal strength. You can see this is a low signal at the top, it's a really strong signal. Why do we get variability in the signal strength? Because the bird is turning around like this, and sometimes the antenna is lined up with the antenna on the back, and sometimes it's not. So when the bird is foraging and moving around, we get a lot of variability. When the bird is resting, we typically get low variability because it's just sitting there, and the signal strength is not changing all the time. So what you can see is that it's foraging at low tide, and then it's resting at the low high tide, and then it's foraging again. So it has chosen to stay and rest at the same place, right? And then here at the next low tide, there is actually a lot of variability, suggesting it's foraging across the whole cycle this time. So two things. One, it stays when it can. Two, it extends its foraging time by being able to remain there. So I think really important things to think about. Those networks need to be close to one another. And this bird has been seen at least a couple of years now. So it must go to northern Russia, or who knows where to breed. Where does it come back to? Millen's Pond. For a couple of years, for 3 years, we've seen it now, right? It just comes back. It loves Miller's Pond. So it's pretty amazing, right, that we see it there again. What are the future plans? Well, we've got 3 antennas that are set up at Port Stephens. We want to now look into estuary movement. We're hoping also to catch up in Port Stephens. So yeah, that's coming your way, HBAC. Last piece of work I'm going to talk to you about. And we talked to you about a study that's relevant to understand this contributing habitats. So sure, roof sites, intertidal flats, foraging areas need to be close together, need to understand where they are. But what about areas that they don't use? I've talked to you about some areas now that don't seem to be so used. You know, what role could they play? Now, one possibility is that we have habitats, estuaries of mosaics of habitats. And it's possible, in an intertidal flat, we know that birds are predators, right? They eat the invertebrates in the mud. But the thing is, what's the vegetation that feeds the invertebrates? Right? The way I like to think about it is if we can protect the intertidal flat, we can say, look, the birds are here at low tide. We really, and they're foraging, and we need to protect this area. The trouble is, if we send the birds to this flat and the restaurant is empty, because the invertebrates are dead, right? Then that's not going to serve its purpose, right? So we need to understand what keeps this a rich invertebrate community that feeds the birds when they come there. Okay, so that's the piece of work I'm going to be telling you about, which is also part of Louise Williams' PhD. Essentially what I'm telling you about is food chains. Pretty simple to understand. You've got primary producers at the bottom of the food chain, then you've got a bunch of consumers, invertebrates that eat that primary vegetation. At the top you've got the predator, the birds that feed on the invertebrates. It's a food chain, pretty easy to understand. And the question I'm asking here is, what are the primary producers, what are the plants at the bottom of this food chain that feed the invertebrates, that feed the birds? Okay, now, that question would actually be really hard because there's not just one plant in the Hunter Estuary, there are many plants, right? There's in any estuary, there's salt marsh, there's fine benthic organic material, which is this layer that you get off the top of the mud, which is a lot of phytoplankton, and then you get mangroves, you get different types of salt marsh. So yeah, which ones feed the invertebrates is the problem that we're dealing with. And that would be really difficult to do if we didn't have something called stable isotopes. Stable isotopes, we've got nitrogen stable isotopes and carbon stable isotopes. What does it mean? It just means nitrogen comes in a heavy form and it comes in a light form. That's basically all you need to know. One's got more atoms than the other. One's heavy, one's light. And the extent of and tissue accumulates nitrogen as you go up the trophic chain. So consumers high up in the chain have more heavy nitrogen than if you're low down. Doesn't matter. One thing is that carbon also comes in heavy and light forms. And the extent to which a primary producer accumulates heavy carbon, a signature of heavy carbon, depends on the photosynthetic pathway of these plants. So-called C3 plants are very impoverished. They have very little heavy carbon, whereas C4 plants have a different photosynthetic pathway. They have lots of heavy carbon. That ratio of heavy to light carbon creates a signature, like your signature on a piece of paper. It creates a signature in the tissue that we can read. And the interesting thing about it is that that signature transfers up the food chain. So we find in the top predator the signature of the plants that separate from the food chain. It's pretty remarkable, right? So if we get tissue from this guy, we can tell which plants are supporting it because we look for that signature here and that signature here, and we match these, okay? So that's how it works. You basically collect all your primary juices, you measure the signature, and then you can model the probability that these different signatures are contributing to the tissue of the predator. That's how it works, they all aspects. So that's what we did. I'm just going to tell you the data on Hunter Estuary. Louise did a comparative study, so we looked at Hunter Estuary. We also looked at a couple of other sites in Western Australia. Really interesting. No time today. Hunter Estuary, what do you do for this study? Well, you collect all your primary producers, all your mangroves, your fine benthic organic material, which is the surface of that mud that we sink into as Fullerton go, and salt marsh plants, etc. You model, you examine in the lab their signature, then you get blood from the bird, a drop of blood from the bird, that's all you need. You need a tiny, tiny amount to get that isotope signature. And then you give it to the experts and they go off and do their modelling magic and they come back with these amazing curves. These amazing curves that essentially tell us which primary producers contribute to the diet. So there is this for Bartel Godwin and for Curly Sandpiper. And the interesting thing that came out of this, so what does it tell you? The more the bars are on the right, the more that primary producer contributes to the diet. That's essentially what you need to know. And this blue contribution here, this blue bar, is this salt cap, so Shorebirds virginicus. And there is a very important signature you can see here in the Curly Sandpiper, although a bit more mixed. Clearly, salt couch is a really important contributor to the diet of both these species. The second most important contributor differs across the two species. So in Bartel Godwin's, you see grey mangroves and your succulent salt marsh plants, which is different to the salt couch, which is a grass salt marsh. And you go with the Sandpiperids, you find benthic organic material. So it's that thin layer of top level of the mud that's really a lot of phytoplankton in it. Okay. So slightly different on their second producers. So how do we interpret these data? Bartel Godwin are typically thought to be a polycrit, so they eat worms. That's what they reported. A lot of worms in their diet if you read the literature. And on the west coast, lots of worms in the mud, and they eat lots of worms. Now, it turns out that Louise has also collected fecal samples from Bartel Godwin, and we find lots of crustaceans in their diet. Because we can look at the DNA signatures of the invertebrates in the fecal samples. And they have lots of crustaceans in their diet. So, very interestingly, also up in Brisbane, they found Bartel Godwin eat lots of crustaceans. Now, this salt couch plant, this Shorebirds virginicus, my colleagues over in the isotope world at the university have actually shown that that is the plant that supports the prawn industry. Right? It also feeds prawns. So it seems as if Shorebirds is it's a very important salt marsh grass that supports the crustacean food chain. So it seems that Bartel Godwin over here on the east coast may be much more crustacean diet than the Bartel Godwins on the west coast, which is, of course, is a different subspecies that some of you wouldn't know. So it suggests there's diet diversification within a species. They can eat different things in different areas. Our curlew sandpiper, they're eating also important salt couch and the fine benthic organic material. We think they are a much more mud forager, so much more polychaete, eating many more worms. That's how we interpret it. What are the key messages? Well, protecting only the mudflat misses the key nutrients. Okay, if you only look at the mudflat and don't protect the salt marsh, you're going to get an empty restaurant. That would be our conclusion. Energy sources differ within and among species, so there's no one size fits all. Sorry, the world is never simple. So we suggest that what is really important is to protect the mosaic of the history, your mudflats, your salt marsh, and your mangroves probably also. And restoration, of course, blue carbon must, when we want to put seed balance with maintaining food web integrity. Okay. So with that, I will conclude by basically saying that I think we have done about 3-4 years of work now in the Hunter, and I think together the work is really increasing the temporal and spatial coverage of the Hunter network. We understand better now how different species are using different areas. We're finding that birds, where they are during the day, doesn't necessarily predict where they are. We're getting some important data about important nighttime risks, because I actually forgot to say here on the I don't know whether I had this on the slide. Yeah, so here, the Bartel Godwin are using this Tomigo site, so I had to come back to this. But we counted there were 280 bi-monthly daytime high tide roost counts at Tomigo in the last 9 years. So 218 surveys during the day at high tide, and Bartel Godwin have only been detected on 22 of them. Right? So you might think Tomigo is not important, but actually it suggests that Tomigo is a really important nighttime site. And while I've got the slide up, I can show you my last conclusion, which was to say, we need to keep that in mind. I might have shown you, that Hexham Swamp, Tomago, they might not be less, Tomago is important for night, but maybe Hexham Swamp, you're thinking, oh, that's not so important, right, you've got marsh happening over there, but it has really important salt marsh. Right? So it is contributing to the mosaic. Even though it might not be used as much, it's still important in the network. And I think with that, I've kind of wrapped up. Yep, importance for primary producers, and I think it's all useful for adaptive management. And with that, I thank the many sponsors of the work. Margaret Flynn has put in an enormous PhD scholarship to support Maxime. I'm very grateful. Local Land Services is a major funder of the work. Cumberland and many foundations have given us funding, and NCRG, Horticulture Services, also supported us, and these people I've already thanked. I throw in a slide for our conference. Please come. We all want you to be there in September. We're really excited about organising this conference. You can follow all our Motus work online. You can go to motus.org and you can see all the sites that are being used by all the species, and there are just some references you're interested in. Thank you very much.